Hello, welcome. Welcome, everybody. Hope everybody's doing well today. Got a couple minutes to go, so give everybody a minute to join in. Thank you for putting your name in chat for me. Feel free to put a nickname in if you want to. All right, very good. We're still missing a couple folks, but I think we'll go ahead and get started. Pardon me while I get my cup of tea all situated over here. Today, we are going to be talking about oxygen requirements. We're going to talk about some laboratory testing we can do to help us learn another characteristic that microbes have, which is the way that they use oxygen or don't use oxygen in some cases. Before we get started, let me ask first if anybody has any questions about anything that they'd like to ask, anything that you've been seeing in lecture or on the quizzes or anything at all that you'd like to ask, feel free, put that in the chat. Remember, today is the day your very first draft is due for your pathogen assignment. I saw that several of you have already turned that in. That's great. If you haven't yet, remember that's due by midnight tonight. So get that into me when you have a moment. It typically takes me a couple of weeks to review all of those and make some comments for you to help guide you as you revise that, that assignment. So I'll be sure to get them back to you in plenty of time for you to then have some time to revise it before the final draft is due. All right. So what I'm going to do is just pull up our homepage in Canvas. If you give me just a moment, just so we can take a look and together review what we have coming up. I'm on the modules page here. You should see that on your screen. And I'm just scrolling down to this week and next week. So of course, this is the module for this week. You had a lecture about fundamentals of biochemistry and then a lecture quiz. I just went through the biochemistry quizzes and you guys did really well on those. So I was very pleased to see that. And then your second lecture for the week is about microbial growth. And then there's another quiz associated with that due tomorrow by midnight. Sorry, due Friday. No, it's tomorrow. I'm right. February 25th by midnight. And of course, in lab today, we'll be talking about oxygen and how microbes use oxygen to grow. Looking ahead to next week, I can't believe it, but we're into March next week. Next week, we're going to talk about how we control the growth of microbes. This week, it's kind of about how they grow. And next week, it's about how humans have learned to control them. And of course, we use lots of different physical methods to control the growth of microbes in our lives. We use things like heat, ultraviolet light, pressure, all kinds of things. And then, of course, we also use chemical things. We use disinfectants and all kinds of agents, chemical agents. And we even use drugs, right? We use drugs to control the growth of microbes in our bodies, antibiotics and such. So that'll be coming up next week. All right. But this week, today, it's about oxygen. So we've just mentioned in class so far that there are microbes, just like there are other organisms that require oxygen. So let me ask, and feel free to type the answer into the chat if you think you know it. Why do humans need oxygen? Why is it that a human being will die if they don't have enough oxygen? What is it that we do with that oxygen? If you think you know, you can type your answer right in the chat. Obviously, you know, if a person, let's see, if a person has some kind of a medical emergency, maybe they have a heart attack and they stop breathing, they're on the clock towards death, right? There's only going to be a few minutes of time left for them before they die. And the reason they die is because they're not breathing and they're not getting oxygen. So the question is, what are they doing with that oxygen? Anybody know? This is a straight up question about metabolism. All right. Again, we're going to talk about this in a couple of weeks in lecture, but the reason that human beings need oxygen to breathe oxygen is because inside of all of our cells, there's a series of metabolic reactions going on that we refer to as aerobic cellular respiration. Now, it's a series of reactions. It's not one reaction. And the first part of this respiration process is the part where glucose is involved. Our cells have a need for glucose, and what they do with the glucose is they take it apart, they break it down. Glucose, remember, has 6 carbons in it. If you remember from chemistry, glucose has a formula of C6H12O6. So there are 6 carbons in glucose. And what our cells are able to do is break those bonds apart. And by the time they finish doing that, and it does take a while because there's several reactions involved, by the time they finish doing that, all that's left behind is carbon dioxide. And you and I exhale the carbon dioxide as a waste product. We can't use the carbon dioxide. So where's the oxygen come in? Well, when we break covalent bonds in molecules, remember, all a covalent bond is a pair of electrons that are being shared between 2 atoms. So in a cell, when a covalent bond gets broken, those 2 electrons are released. They're released from the bond. And our cells are able to harvest them, to sort of grab onto them and hold them temporarily so they don't get lost as heat. And what our cells do with those electrons is they feed them into another series of reactions called the electron transport chain. And what happens is the electrons go through these reactions and the energy that's in the electrons gets removed, it gets harvested. That energy that comes from those electrons is going to be used to build that very important molecule called ATP. But there's one other thing that has to happen. Those electrons have to go somewhere. We've taken energy out of them, but now we need to find them a home, because remember, nature doesn't like electrons just floating around. It's very unstable. Nature wants electrons paired up in molecules. So something has to accept those electrons that are passed down this electron transport chain, this series of reactions. Something has to be willing to take them, to accept them. And that something is oxygen. So what we say is the final electron acceptor molecule, the final electron acceptor molecule at the end of the electron transport chain is oxygen. Oxygen takes the electrons. It also takes some hydrogen, and it makes water. And that's how that whole series of reactions finishes up. Breaking apart glucose, setting the electrons free, taking the energy from the electrons so that it can be used to build ATP, and then taking the electrons and giving them to oxygen so that water can be formed. We are oxygen users. We are aerobic organisms, so we do aerobic cellular respiration, and there are lots of microbes that do that too. If we don't have oxygen, we'll die, because if we don't have oxygen, the electron transport chain has nowhere to send those electrons, and it will shut down without oxygen. And if it shuts down, that means no more ATP can be made. And our cells don't store a whole lot of ATP. They store a few minutes worth. So if we stop breathing and we stop getting oxygen delivered to our cells, we're on the clock. We've got a few minutes left. Think about one of the things you learn in emergency medicine, in first aid, for example, when you take your CPR classes, the ABCs of CPR. That airway and breathing is right at the top, right? Got to get the airway clear, and you have to get that person some breath. You've got to breathe for them or apply oxygen, because that is a molecule that we cannot live without for very long. And there are lots of bacteria that are the same way we are. But there are lots of bacteria that are not like us. Remember, they're much more diverse than we are, metabolically speaking. There are many bacteria who use a process that's very similar, but it doesn't involve oxygen. There's a process called anaerobic cellular respiration. So the reactions are all very similar. Oxygen gets broken down into carbon dioxide. All of the electrons get released from the covalent bonds. Those electrons get passed down the electron transport chain. Their energy is used to build ATP. But at the bottom of their electron transport chain is not oxygen. It's another final electron acceptor molecule. So they're still doing respiration. They're still doing cellular respiration, but they don't use oxygen as their final electron acceptor. Some microbes, some bacteria can use things like nitrate and sulfate and carbonate. Those are all oxygen-containing molecules, but they're not oxygen. So they do the same type of cellular respiration reactions. They just use a different final electron acceptor molecule. Now there is a third set of reactions to generate ATP that bacteria can use, and that's fermentation. Fermentation is not nearly as efficient as cellular respiration is. In other words, for each molecule of glucose that the organism breaks down, you get a lot fewer ATP at the end if you're fermenting. But there are some microbes that use fermentation as sort of a backup pathway. If they don't have their final electron acceptor molecule around, they can ferment instead. There are other microbes that use fermentation exclusively. It's the only pathway they use. So we see all kinds of combinations of aerobic cellular respiration, anaerobic cellular respiration, and fermentation in microbes. And they're all a little different, but they all generate some ATP for the cell. So how do we figure out what kind of metabolic reactions these microbes are doing to make their ATP? How do we figure out if they're doing aerobic cellular respiration, anaerobic cellular respiration, or fermentation, or some combination of those? Well, there's no one test we can do. We have to do a couple of different tests. But when it comes to oxygen, we can do some fairly easy tests that will give us a really good idea of whether or not the microbe we're dealing with uses oxygen, doesn't use oxygen, or uses it sometimes and not other times. Because again, they're very diverse, these microbes. They're able to do things that you and I just aren't able to do. So that's what we're going to talk about today. We're going to talk about oxygen. Now, I'm going to go ahead and pull up our slides. And that's what you should be seeing now. You should see a title slide that says Oxygen and Growth. Let's talk about the objectives that we have for today. We're going to talk about 5 different categories of oxygen usage for microbes. And we're going to be talking about bacteria really today. So there are 5 different categories that bacteria can fall in terms of how they use oxygen or don't use oxygen. So that's the first thing we'll do. We'll talk about each category and what the names mean. Then we're going to look at a type of culture medium that is used to help us figure out which category an unknown microbe falls in. This special medium is called thioglycolate medium. And this is the first medium that we'll look at that has that semi-solid consistency. We're going to inoculate the medium and we're going to let microbes grow in it. And we're going to look for a pattern of how they grow in the tube to tell us which category they belong to. We're going to talk about a couple of culturing methods. So not necessarily media, but methods that we use when we want to culture an anaerobic organism, an organism that we know doesn't use oxygen. We're going to talk about a test called the Catalase test. Catalase is an enzyme. And by the way, if you don't know this trick, when you see a term like catalase that ends in ASE, that tells you it's an enzyme. And remember, all an enzyme is a protein that is capable of catalyzing a chemical reaction. In other words, making the chemical reaction occur quickly, more quickly than it would otherwise. So we'll talk about what catalase does. We'll talk about how we do this test. This is a very simple test. And how we identify a positive or a negative. And then finally, just like last time, we'll do a little experiment. We're going to try to figure out the oxygen category, again, from these 5, for 4 different bacterial species. So I've talked for a few minutes about the ways that cells make ATP. human cells and microbial cells like bacteria. I just want to say a couple of things about this important molecule called ATP. This complicated looking structure up here, this is ATP drawn out as a molecule. You can see that there's a ring structure in the center here that has an oxygen in the midst of it. And then you can see something off to the side here, two other rings that have a lot of nitrogen in them. And then you can see 1, 2, 3, what we call phosphate groups. And that's kind of what makes ATP an energy currency. Cells can break off this outermost phosphate group and release the energy that's in the bond right here. Remember, covalent bonds, they're two electrons, a pair of electrons that are being shared between two atoms. And when the bond is broken, energy is released because the electrons are released. And energy is carried with electrons in living cells. So when we build a covalent bond between two atoms, we are storing energy. And when we break that bond, we are releasing energy. So cells use ATP as a little storage molecule for energy. And they break off this outer phosphate group when they need a little bit of energy to do chemical reactions. All living cells need a constant supply of ATP. And that's because cells do not store large amounts of this. They make it as they need it. This is what ATP stands for, adenosine triphosphate. Tri meaning three phosphate groups are on this molecule. So there's the 3 P's, the 3 phosphorus atoms. And what makes it a phosphate group is that it's got oxygen around it. So each one of these are phosphate groups. We said there are 3 complex metabolic pathways that are used to make ATP in microbes and other organisms. And we can test microbes in culture to see which oxygen category they fall into. And we can do additional tests to determine which of these pathways they're able to use. Now, why would we care about that? Well, number one, it can help us identify a microbe by knowing what kind of metabolism it has. But if we want to grow a microbe in the laboratory, we also need to know what kind of oxygen requirements it has, or if it doesn't want oxygen around it. That will help us culture organisms if we understand them better in terms of their oxygen needs. So let's talk about these 5 different categories that organisms fall into. And again, today we're talking primarily about bacteria. The first category is the obligate aerobe. That word obligate kind of gives it away. These are oxygens that require, these are organisms that require oxygen. They require oxygen, again, to make their ATP. So human beings are obligate aerobes, and so are a good number of bacteria. These bacteria are going to use aerobic cellular respiration, as we said, with oxygen as the final electron acceptor molecule. But a lot of them can do fermentation as well. It varies by the species. And these are organisms that tend to have this enzyme called catalase. Now let's talk about catalase for just a moment. Catalase is an enzyme that can break down hydrogen peroxide. It takes the hydrogen peroxide molecule, and down at the bottom here, I've got the formula for you, H2O2 is hydrogen peroxide. Hydrogen peroxide is toxic. That's why we often use it when we're cleaning wounds, right? It'll help kill any bacteria or other microbes that are in the wound. Hydrogen peroxide is toxic, and hydrogen peroxide gets generated inside cells that use a lot of oxygen. In other words, it's sort of a byproduct of all that oxygen that's in the cell being used for metabolic reactions. You know, we think about oxygen as being this really great molecule because, of course, we need it to survive. But oxygen is not safe in large amounts over the long term. Oxygen is actually quite toxic. Those of you who work in the healthcare field know that when we put a patient on 100% oxygen for some reason, we don't leave them on that any longer than absolutely necessary. We want them to be breathing room air. And that's because too much oxygen can become toxic to your cells, because oxygen can be converted into other molecules that are not safe for our cells, like hydrogen peroxide. So in obligate aerobes, because they tend to have a lot of oxygen in their cells, and therefore they also tend to generate hydrogen peroxide in their cells, they need an enzyme to break it down. And that's what Catalase does. Catalase takes hydrogen peroxide and makes water and oxygen gas with it. I was just looking to see if I have the formula for you, but I guess it's later on. So you might imagine that if you're a microbe that doesn't use oxygen, you probably don't need any catalase around, right? And you'd be right, because the second group are the obligate anaerobes. These are organisms that not only can't use oxygen to make ATP, they use a different molecule as their final electron acceptor. But to them, oxygen is actually toxic. If these microbes get exposed to oxygen in the air, it will kill them. So these are organisms that use anaerobic cellular respiration and or fermentation, depending on the species again. And they don't have catalase. They don't need catalase because they don't need to have oxygen around in their cells. Oxygen is toxic to them. So there's no need to break down hydrogen peroxide for them. Now let's look at the third group. These two first groups, I think, are kind of straightforward. They have an absolute requirement for oxygen, and oxygen is toxic. But the rest of the groups are somewhere in between. The third group are called aerotolerant anaerobes. So remember, anaerobe makes you think no oxygen. But they are aerotolerant anaerobes. So just like the obligate anaerobes, these organisms cannot use oxygen to make ATP, but oxygen is not toxic to them. So they can be exposed to oxygen. It's not going to hurt them. They just don't use it. They don't use it to make ATP. They too tend to lack that catalase enzyme. They just don't need it. In fact, it turns out that they don't even have the system inside of their cells that even generates hydrogen peroxide. So not only do they not have catalase, but they don't even know how to generate hydrogen peroxide. So it's not a problem for them. Now, just like the obligate anaerobes, the aerotolerant anaerobes can use anaerobic cellular respiration and or fermentation. But most of them use fermentation. So these are organisms that we refer to as pure fermenters. That's the only metabolic pathway a lot of these organisms use. Now, here's something important to know about the aerotolerant group. They grow best in a little bit of oxygen. Remember, they're not using it for their ATP production, but they use it in other ways, and they actually prefer to be in a low concentration of oxygen. A low concentration of oxygen will kill an obligate anaerobe, but the aerotolerant ones actually like it. They like it best. Now I've got the fourth and the fifth groups on this slide. Number 4 is the micro aerophile. Micro meaning small or low, aero referring to air or oxygen, and phile means love. Remember, philic, hydrophilic means water loving. A micro aerophile loves oxygen, but only loves it in small amounts. So this is an organism that requires oxygen to make ATP, just like the obligate aerobes. But this organism needs its oxygen at a reduced concentration. A reduced concentration. Now, here's a question. How much oxygen is in the air? How much oxygen is in the air we breathe? Anybody know the percentage? It's just about 21%. What's the rest of what else is in our air if it's not oxygen? What are we breathing? Anybody know what the other big element is in our air? Remember, there are 4 key biological elements, right? Oxygen, nitrogen, carbon, hydrogen. What's the element that makes up most of our air? Anybody? It's actually nitrogen. Most of what's in your air is nitrogen. About 21% oxygen in the air. Just right for us and for some of the microbes. But some microbes need a lower level of oxygen than that. All right. Microaerophiles grow best in a low concentration of oxygen and a higher than normal concentration of carbon dioxide. So air, the air that we breathe, has less than 5% carbon dioxide in it. They prefer somewhere between 5 and 10% carbon dioxide. So they like a little more carbon dioxide than what's in the air and less oxygen than what's in the air. Now, you might ask yourself, gosh, how do you create those conditions if you want to grow a microaerophile in the laboratory? How do you create that? It turns out there's a really basic device that you can purchase. It's called a CO2 jar. In the old days, scientists used to take a jar, like just a regular glass jar that has a tight fitting lid. They would put an agar plate or a broth tube, an inoculated plate or tube, into the jar with a lighted candle. And then they would close the lid. And what ends up happening is the candle will burn up a lot of the oxygen, right? And generate some carbon dioxide. So you create a little micro environment in there. But nowadays you can actually purchase a commercial packet that will generate some carbon dioxide and consume some oxygen. It's not taking all of the oxygen away. Remember, microaerophiles want some oxygen. They just don't want a lot of it. Now, the last category on the list is called the facultative anaerobe. So this is yet another anaerobe. The term facultative is the important term here. This term means an organism that can sort of adapt to its environment. Facultative anaerobes grow best with oxygen, but they don't require it to make their ATP. These are often organisms that can do a whole combination of pathways, metabolic pathways. They can sort of switch from one to the other. So when there's oxygen around, they'll do aerobic cellular respiration, just like you and I will. But if there's no oxygen around, well, they'll switch over and they'll do anaerobic cellular respiration, or they'll do fermentation. They are the most adaptive group of microbes. And notice this down at the bottom. Most of the bacteria that we encounter are in this category. Most of the bacterial species that we encounter are facultative anaerobes. Very, very adaptive, very able to adjust to changing conditions. So I mentioned that we use a special kind of medium called thioglycolate to look for which category these microbes belong to. And that's what this slide is about. Obviously, these are just drawings, but they're a representation for what microbes look like when they grow in thioglycolate. Now, we do call it a broth, and it is semi-solid. It's not as solid as agar is, but it's thicker than regular water is. I like to think of it as having the consistency of, something along the lines of maybe a thick soup or something. When you pour thioglycolate, it's thick. It's not like regular nutrient broth. So the way they make thioglycolate, they take just a basal medium. Remember, that's a very basic defined medium. And they add a little bit of agar to it, a little bit of powdered agar to thicken it up. Now, why they add the agar is because we're going to let oxygen from the air interact with this medium. And the oxygen, as it comes into the tube, it's going to be able to dissolve into the upper parts of the tube, into the liquid at the very top of the tube. But the lower parts of the tube, it won't be able to dissolve into. And that's because of the agar. So what you set up in one of these tubes is two zones. You set up an oxygen zone, and you set up a zone that doesn't have any oxygen in it. The very top part, and it's generally about a quarter to maybe a fifth of the volume, is where the oxygen is going to be. So we call this the oxic zone. And then the rest of the tube is called the anoxic zone, or without oxygen. You can buy thioglycolate pre-made, and most of the labs I've worked in buy thioglycolate because it's kind of a pain to make. You can even add an indicator dye in here. Remember, we said indicator dyes change color when pH changes. So if there's an indicator dye in the tube, even before you inoculate it, the media is going to turn pink in the oxic zone, and it will stay this regular beige color in the anoxic zone. So some people like that because they can clearly see where the oxic zone is and where the anoxic zone is. So we take these tubes and we inoculate them. We take some cells from culture with a sterile loop, and we put the loop down into this sterile thioglycolate broth and we swirl it around a little bit. And then we put the tubes in the incubator and we let the microbe grow. And the pattern of growth, that's what you're seeing with the red. The pattern of growth is going to tell us what category the microbe belongs in. So obviously microbes aren't red, but remember, broth is typically very clear. It's very transparent. Even with the agar in this, it's still quite clear when it's uninoculated. Where the microbes grow, the thioglycolate broth is going to get cloudy. And sometimes it gets very cloudy, like it turns almost a white color. There's so many cells growing in it. It depends on how long you leave them in the incubator. So let's take a look at these categories. This one over here on the left, this is the way obligate aerobes grow in thioglycolate broth. Remember, these organisms require oxygen to make their ATP. They will die without oxygen. So, no surprise, they are limited to growing in that upper portion of the tube where the oxic zone is. In comparison, we have the obligate anaerobe. This is the one where oxygen is toxic to them. No surprise, they grow way at the bottom of the anoxic zone. Even though we call this whole region here, from here down, we call the whole region an anoxic zone, there is no strict, hard cutoff from where the oxygen is and where it isn't. In other words, there's a good amount of oxygen in the top, again, 20 to 25% of the tube. And as you go down from there, there's less and less oxygen. So by the time you get to the bottom, there's no oxygen. And that's why they grow there, right at the bottom. Now take a look at the other categories. Remember, the next one on our list was actually aerotolerant. That's this one at letter D. And if you remember when we talked about aerotolerant, we said they can't use oxygen to make ATP, but it's not toxic to them. They tolerate it. And in fact, they grow best in low oxygen conditions. So if you look at this tube, first of all, you'll notice this strange green square right here. I'll show you the way the artist drew the tube. The artist showed that the cells are evenly dispersed all the way up and down this tube. But in my experience, this is not accurate. In my experience, you just don't see growth up at the top, at least not in any significant amount. You'll see it through the rest of the tube, but not way up at the top. Aerotolerant organisms, as I've got written here, tend to grow up to the lower regions of the oxic zone. Okay, they can grow without oxygen, remember, but they can't, they would rather not be in high oxygen levels. So they just don't tend to go to the top of the tube, in my experience. Here's our micro aerophile. These are probably the easiest ones to see when you look. Oh, I mean, obviously these two are quite easy. But this one is very strange because it's at the top of the tube, but not the very top. Remember, a micro aerophile requires oxygen. It's got to have it, but it likes it in low concentrations. So they grow right here in the lower portion of the oxic zone. Obligate aerobes can grow all the way through the oxic zone. Microaerophiles are limited to the lower half. And finally, we have our facultative anaerobes. They too can grow all the way through the tube, just like the aerotolerants can. But if you remember, we said facultative anaerobes grow best with oxygen. So when we look at the tube, we're going to see most growth up at the top, like the obligates. But they can also grow without oxygen. So they grow all through the tube with most of the growth at the top. Now, another note, this is important here. Depending on how long you leave these tubes in the incubator, once you inoculate them, if you grow a lot of cells in these tubes, what tends to happen is they clump together. So you're going to get a clumping that happens all the way through the oxic zone or way down at the bottom of the anoxic zone. And then in a tube like this one, what you'll see is a big clump of cells up at the top. And then you'll see these strange little strings hanging down. Those are cells clumping together. Remember, we said bacteria, they like to associate with each other. They don't like to be all alone in culture or otherwise. So you don't see them nicely and evenly dispersed like this. Instead, you tend to get these weird strings hanging down through the media. Same with this tube. All right, so a lot of information so far, but does it make sense to you what we've been saying so far? The thioglycolate is a useful media because it can create these zones in the tube where oxygen is plentiful and oxygen is not plentiful. I forgot to mention it, but thioglycolate also contains additional chemicals, something that we call reducing agents, which also help control the amount of oxygen in that lower portion of the tube. Very, very useful media if you know what the patterns look like. All right, I'm going to jump back in. We said at the beginning of the lab today that we were going to take a look at this catalase test, and I told you it's very easy. This is the easiest test you'll ever do in the lab. And again, whether or not a microbe has this enzyme is a characteristic for that microbe. It helps us identify them. So this is a test that we do frequently in the laboratory. To do a catalase test, all you need is a glass slide, a bottle of hydrogen peroxide, and something like a swab to mix it up with. So Catalase is the enzyme that breaks down hydrogen peroxide. And hydrogen peroxide is just a byproduct, something that oxygen sometimes gets turned into in living cells. It's toxic. It's toxic to all cells. So hydrogen peroxide in the presence of this enzyme, catalase, will be broken down into water and oxygen gas. The way we do the test is we take a little bit of culture material, preferably from an agar plate. So we pick up a few of the colonies from an agar plate, and we just smear them onto the surface of a glass slide, as though we were making a smear, but a thicker smear than what we would normally make. Then we're going to add a drop of hydrogen peroxide on top of those cells, and we're going to watch. It happens really fast. If the cells make catalase, they're going to start doing this. They're going to start bubbling. Those bubbles are just oxygen gas. Inside the cells where we can't see, the hydrogen peroxide is being broken down. It's being broken down into water and oxygen gas, and the oxygen gas just gets liberated like this. And you'll get this nice foamy, bubbly little bundle right here. If they don't have catalase, nothing happens. You just put a drop of hydrogen peroxide on. You might mix it around a little bit. Nothing happens. There's no catalase, so they can't do anything with it. So a very satisfying test to do because you get to see it right away. Hey, here's a question. Eukaryotic cells like you and me, where do we keep our catalase? Because we do make catalase. Anybody remember which organelle in a eukaryotic cell, like a human cell, breaks down hydrogen peroxide? The name of the organelle.
Is it the peroxisome?
That's exactly right. I love it when something is named appropriately. So the peroxisome is the organelle that breaks down hydrogen peroxide, as well as a few other things. But it's the peroxisome that holds on to the catalase enzyme in our cells. So as hydrogen peroxide is generated as a byproduct, the peroxisome will take care of it. The peroxisome degrades it and makes it go away. Good. All right, let's talk for just a minute about what we do in terms of culturing when we know we have an anaerobe. Remember, if we have an obligate anaerobe, we've got an organism that can't be near oxygen. Oxygen is toxic to it. So there are a couple of ways that we can grow anaerobes in the laboratory. And again, I'm talking about obligate anaerobes here primarily. Let me fix that. So the first thing you have to do is just, you've got to get the oxygen out, right? You got to remove oxygen so that they can be in an environment where there's no oxygen. There's a really clever, again, a jar that you can buy commercially called a brewer's jar or a brewer's anaerobic jar. This jar has a tight fitting lid, so you can put, an inoculated agar plate in there, or you can put a broth tube in there. But remember, you've got to get the oxygen out. So the way this happens is you buy these gas packs. They look like juice pouches, the juice pouches that have an aluminum foil type of container. These packs have hydrogen gas in them. And what you do is you put the hydrogen gas packet into the brewer's jar and you basically open it. Then you shut the lid. And what's going to happen is that hydrogen gas is going to combine with any oxygen and all oxygen inside that jar. And it's going to create water. So when you use a brewer's jar, you get this condensation on the inside of the jar, and that tells you that things are working. We also add in one of these indicator dyes. It also comes in a little package, but these dyes are there just in case some oxygen gets in. We'll know because this packet will turn color if any oxygen gets into that container while it's incubating. So we do something actively to remove the oxygen from the jar. And then we make sure we keep watch just in case any oxygen accidentally gets in. You then just take this brewer's jar and you put it in the incubator in that nice, warm, dark place, and your anaerobic organism will grow for you. So the brewer's jar is one method we use, but we can also use, again, some kind of culture medium that can remove oxygen. And again, the term we use is reducing, a reducing culture medium. That's what thioglycolate broth is. Thioglycolate broth is a culture medium that can control how much oxygen is in it. And you get those zones. You get an oxygen-rich zone or an oxic zone. And then down in the deep regions, you get an anoxic zone. And as I said, you can put an indicator dye in this broth, and it will show you where the oxic zone is. It will show you where the highest concentrations of oxygen are. up at the top of the tube. Now, there's a third couple of things that I have at the bottom in parentheses because these are not common. Thioglycolate broth and the brewer's jar, very, very commonly encountered in microbiology labs. But if you happen to be a lab that works with anaerobes a lot, and those are usually research labs, you can buy an anaerobic incubator. So it's an incubator that seals and then has the ability to pump the oxygen out. Those are very expensive, as you might imagine. There are also what we call glove boxes, and that's what this item is. This glove box, as you can see, there are 2 openings here that have gloves attached. So I can put my hands through these openings and into the gloves, and I can work in this chamber. There are valves up at the top that you would attach to pumps to get the oxygen out of this chamber. So glove boxes and anaerobic incubators, not common. You're not going to see these in many labs, but they are available if you're trying to grow anaerobes. Here's what the brewer's jar looks like. It's got a really tight fitting cap on it, and you have to tighten up this thumb screw up here to really get that cap seated. And these are the little packs that come with it. The silver pack, this is the one that has the hydrogen gas in it. And then you also have this white pack that has the indicator dye in it. It's really frustrating when you try to grow anaerobes and you're not, you haven't done it very often. And you set up your brewer's jar and you put it in the incubator and you come back 24 hours later all excited, because you've grown your anaerobes, and this indicator pack has changed color and turned like red or pink. And you're like, oh no, oxygen got in. The seal wasn't tight enough generally. That's what that means. Some oxygen gets in and that'll kill the cells. So it can be very frustrating to try to grow these anaerobes. This tube right here, this is what a thioglycolate tube looks like when you buy it commercially. Again, it just looks like broth. But if you tilted this tube side to side, you'd see it's a little bit thicker than regular broth. And this over here, this other image, this is a thioglycolate tube that has that indicator dye added to it. And it literally does this. The very top of the tube, the liquid will turn pink. That's where the highest concentration of oxygen is. That's the oxic zone. There is a little bit of oxygen down here, but not much. And there is just none down here. Thioglycolate broth is a very commonly encountered media in microbiology labs. We use it a lot. It's not particularly expensive. As I said, most people purchase it because it is kind of a pain to make. And it can give us a lot of information about the oxygen characteristics of an unknown microbe. All right. Well, let's start into our experiment now. Just like last time, what we'll do first is we'll talk about which microbes we're going to use, and then we'll talk about what media we're going to use, and then we'll look at some results. So today, on this slide, this is a list of the bugs we're going to use. The first, these are all bacteria, I should say. And it's interesting to note that all of these can be found in the human intestine and down in our GI tract. So they can be all part of our microbiome. But there are some strains of all of these that are pathogens. So again, a very common feature of bacteria. There are good E. coli and there are bad E. coli. So let's look at top to bottom here. The first one is called Clostridium sporogenes. That's how you pronounce that strange looking word. Clostridium sporogenes is a gram-positive rod. So we haven't seen too many gram-positive rods. This one can be found not only in our gut, but it can also be found out in the world. It tends to be in soil, or you can also find it in sediment, in bodies of water. So down at the bottom of the body of water. This is an organism that can survive in certain concentrations of seawater. So brackish water, as we call it. Soil and also in the gut. This is what Clostridium sporogenes looks like when it's gram stained. As you can see, it's purple when it's gram stained, so it's gram positive, and it's rod shaped. See that? Remember too that from lecture, there are two genuses, or what we call genera. There are two different genus of bacteria that are pathogenic at times that can make spores. Bacillus and Clostridium. Those are the two genuses or genera that you should know, because they're capable of making spores. There are lots of other bacteria that can also sporulate, but we don't tend to encounter them. They live out in the environment. But the two that we encounter in our body as pathogens sometimes are Clostridium and Bacillus. Now, here's our second one, Enterococcus faecalis. We've seen this one before. A gram-positive coccus. This one you could almost call a coccobacillus. I don't know if you remember this term, but because I told you this is not a very common shape. But there are some bacteria that really aren't true cocci. They're more oval than they are round. See that? It's not long enough to be considered a rod. It's considered a coccus, but look at how long it can be, or how, I shouldn't say long, how oval shaped it can be. So you would not be wrong calling this a coccobacillus. In other words, halfway between a coccus and a rod. This one too lives in our gut. You can also find Enterococcus faecalis in the mouth and also in the reproductive tract, particularly in the vagina. Part of the flora. Here's our old friend E. coli, gram-negative rod, found in the GI tract. You've seen E. coli before, but here's another slide with E. coli on them. This is only 40x, so it's a little bit hard to see, but it's very clearly gram-negative. You can see 1, 2, 3 of the rods magnified right here. Remember, E. coli are rods, but they're stubby little rods. They're short compared to most other rods. And finally, we have another organism. This one has another very strange name. This is called Alcaligenes faecalis. And like E. coli, this one is also a gram-negative rod. It can be found in the GI tract again, and it can also be found out in the world in soils. So similar to the Clostridium, you can find Alcaligenes out in the environment in soils and sediments. So 4 organisms, all 4 of them can be encountered inside the human body in the GI tract. And we're going to try to figure out what oxygen category they belong to. All of these cultures were confirmed to be alive before we used them, so we know they're okay. Now let's look at the media we're going to use. We're going to use thioglycolate broth tubes, although ours do not have the indicator dye in them. We're going to use nutrient agar plates, and we're going to use the plates so that we can compare growth in a brewer's jar with growth in room air. Okay, so we're just going to grow them on a basic agar plate, but we're going to expose them to different amounts of oxygen. And the last thing we'll do is catalase testing. All right. So we're going to make some predictions about what we think we're going to find growing in our thioglycolate broth and on our nutrient agar plates. Remember, we have some organisms of those 5 categories that can live and grow in the presence of oxygen. Some of them have to have the oxygen. Some of them tolerate the oxygen, right? Obligate aerobes, they can grow in oxygen. Facultative anaerobes, yup. Remember, we said these are the ones that can switch back and forth. They prefer oxygen. They grow best in oxygen. But if there's no oxygen, well, they can grow in that too. Aerotolerant and microaerophile, remember, these are the 2 groups that it's okay for oxygen to be around, but it has to be in low concentrations. So if they're just exposed to air, they're going to grow, but they're going to grow poorly. They're not happy when they're at 21% oxygen. It's just too much oxygen for them. But oxygen is not toxic to them. It's not going to kill them outright. Now you notice that the obligate anaerobe is not on the list, right? Because an obligate anaerobe cannot grow in air or oxygen. Oxygen is toxic to that organism. So 4 of our groups can grow in the presence of oxygen. So what can grow on nutrient agar plates? That's what the Na is. In a brewer's jar where there is no oxygen. Well, the obligate anaerobe for sure can grow in there, but so can that facultative organism. Remember, we said it prefers oxygen, but it doesn't have to have it. So that's on the list. And we can put aerotolerants on the list as well, but they're going to grow poorly. Let me pull them back up for just a second so you can take a look at this and review it. Aerotolerants don't use oxygen to make ATP. They tolerate it. So if there's no oxygen around, they're going to be fine, but they grow best when there is oxygen around. So if I put them in a brewer's jar where there's no oxygen, they're not going to grow well. All right, they're not going to grow well. They're going to grow poorly. So these are how we would break down the groups into yes or no on the oxygen. And then let's also think about the catalase test. Who should have a positive catalase test? Well, obligate aerobes will. Facultative aerobes also will, and so will the microaerophiles. You'll notice that these are the 3 that either require oxygen or prefer oxygen. Oxygen in any amount, because of course the microaerophile prefers it in low concentrations. They require it to make their ATP. We expect them to have the catalase enzyme. Who should have a negative catalase test? The other 2, the obligate anaerobes and the aerotolerant organisms. We don't expect them to have catalase. Remember, we're going to look at these patterns in our thioglycolate tubes, but we're going to remember that it's not going to look as perfect as this because growing cells tend to clump together. All right, are you ready for some results? Let's look at the nutrient agar plates first. I don't have pictures of these for you, I apologize, but they just didn't come out very good. My pictures were too dark. So on the nutrient agar plates that were incubated in the air, in the room air. Here's the results for our 4 organisms. The Clostridium sporogenes had no growth at all on it. The Enterococcus faecalis grew, but it grew very poorly. In other words, there were colonies on that plate, but they were really small, not as big as they should have been. The E. coli grew great, normal size, regular colonies. The alkaligenes grew great, normal size, regular colonies. Now let's look at the plate that we put in the brewer's jar. Clostridium sporogenes grew great, nice normal size colonies. The Enterococcus faecalis grew great, nice normal size colonies. The E. coli grew, but it grew very poorly, little colonies. And the alkaligenes did not grow at all. So just from these results, do you think you have any obligate organisms? Do you think we have any obligate aerobes? I'll pull up the slides again. Obligate aerobes.
I'm gonna say. Yes.
Which one?
Let me say correctly.
You can say. You can say the initial.
Can we say the A. faecalis?
Yeah, that's fine. A. faecalis. That's right. It's a terrible word. It's alkaligenes. Terrible
Yes.
word to pronounce.
And that was 3 pages back in notes. So.
That's right. Yes, this organism grew really well in oxygen and was not able to grow at all when there was no oxygen. So that's a really good clue that this is an organism that must have oxygen or it cannot grow. Now, what about an obligate anaerobe? What do you think? Kind of the opposite, right? An organism that cannot grow in the presence of oxygen, but grows well when there is no oxygen. Have we got one of those?
Is it the first one, the C?
Yeah. C. sporogenes. Yeah, that's exactly right. This Clostridium organism, no growth in room air. Oxygen is toxic to this bug. Will not grow in room air. When you put it in a jar where all the oxygen has been removed, it grows great. So we already have a good idea that we have an obligate anaerobe and an obligate aerobe in this group. Now, think about A. faecalis for a minute. An obligate aerobe must have oxygen. Where would you expect to see it growing in the thioglycolate tube?
At the top.
At the top, right? Oxic zone. It should grow all the way through that oxic zone. We should see a nice sort of clump of cells growing at the top of that tube. Very good. How about the Clostridium? Where is that Clostridium going to want to grow in that thioglycolate tube? Oxygen is toxic to it.
Bottom.
All the way at the bottom. All the way at the bottom. That's right. It's not going to grow all the way through the anoxic zone. It's going to head for the bottom of the tube. Very good. This is our reminder of what can and can't for oxygen. So, you know, these obligate organisms, they're the first ones to come to mind when we see no growth in one of the conditions. All right, so here are our thioglycolate tubes. We'll look at a couple of them at a time. Here is the Clostridium sporogenes tube. Now, this is a good lesson in how things don't always look like the textbook, right? Here's the thioglycolate. Remember, the oxic zone is going to be the top 20 to 25% of the tube. We don't have any indicator dye in here, so we just have to eyeball it. We expect Clostridium, like we just said, to grow way at the bottom. But we didn't get much growth, did we? This right here, that's how much we got. We got a little clump. When I say a lot of growth, I would expect this whole bottom region to be full of this cream colored, whitish yellow material. But instead, I just got a little clump growing. So it could be that, you know, something wasn't right, that I didn't do a very good job protecting the cells from oxygen before I used them to inoculate the broth. We got growth, but we didn't get much. But it's right where it should be. It's right at the bottom of the tube. So we have more evidence that Clostridium sporogenes is an obligate anaerobe. I'm going to skip ahead for just a minute so we can look at alkaligenes. Remember, we said alkaligenes is an obligate aerobe. And there she is. She is growing in a big clump. You don't see individual cells or anything close to that. You just see this big clump of material growing. This is all cellular. And it's right in that oxic zone. Classic for this. It cannot manage down here. There's not enough oxygen for it to survive. Now let's look at the other ones, because these ones are in the categories that aren't quite as easy. Let's look at the Enterococcus first. Let me get this nice and big so we can see it. You can definitely see a mass of cells, a big clump of cells right here. And this is what I mean by the tendrils, the strings that kind of hang down. Cells clump together in these tubes. They don't stay separated from each other or suspended in the broth. So the way you read this, when you see a tendril like this, is that cells are able to grow all the way through this, right? They're able to grow at this level and this level and this level and this level and down at the bottom. But most of the cells are up here. You'll notice they're not all the way up to the very top, are they? There's part of the oxic zone here that there's nothing growing in. So we have a preference right around here, but we have the ability to grow all the way down. Who does this remind you of? This is a hard one to interpret. In fact, let me do this first. Let me pull up our categories.
Well, when I looked at it, it looks like what you described as a facultative anaerobe.
Yep. Facultative, remember, can grow all the way through the tube, prefers to be in oxygen, but can grow all the way through the tube. Here's why it's not facultative. Let me get down here again. A facultative would have grown all the way up to the top. This organism is avoiding the very top of that tube. Did we talk about a category that likes to avoid the highest concentrations of oxygen, but is capable of growing all the way down?
The obligate anaerobes?
No, remember, obligate anaerobe, we're only going to get at the bottom. So it's not.
Oh, pick me. Aerotolerant.
Very good. Pick me. Very good. Aerotolerant. That's exactly right. Let me pull it back up for us. Look at the aerotolerant description. It doesn't use oxygen, but it tolerates it. Oxygen is not toxic. It grows best in low O2 conditions. All right, it grows best in low O2. We've got no growth at the very top where the most oxygen is. We've got less oxygen here, and of course, less and less and less and less oxygen all the way down, but there's less growth as we go down as well. So this organism is growing best right here, but it can grow all the way down. This is aerotolerant. And here's our old friend, E. coli. We got a couple of very interesting looking tubes for E. coli, which is why I put a couple pictures in here for you. Look at these tendrils. They're so bizarre. They look like they're hanging in the middle of nowhere. There's agar in here, which is why they're able to be suspended like this. Very, very strange. Over here, you can also see a big clump here, and there is a tendril right here. If you're seeing a sort of an opaque line, there is a tendril right here. Okay, so when we look at this one, what do we see? We see a lot of growth up top on this tube, oxic zone again, but the ability to grow to a lesser extent all the way down. That's the tendril. Over here, there's a little tiny bit right here that we don't see anything growing, but it's not very big. Most growth is right here, and there's tendrils hanging down. Could this be another aerotolerant, or do you think it's something else? Look at this tube.
Is it the micro aerophile?
There's one other one, Vicky. Microaerophile does not like high concentrations of oxygen, so it would not grow up here. And microaerophile cannot grow down here. But there's one other category that this meets. Most growth in the oxic zone, but able to grow all the way down. Anybody?
Facultative anaerobic.
That's your facultate, right? That's a facultative anaerobe. Most of the growth is going to be up at the top because that kind of organism prefers oxygen. If there's oxygen around, it's going to do metabolic reactions with oxygen. It's going to do aerobic cellular respiration. But if there's no oxygen around, it's still okay. It'll grow. It's not going to grow as well as it does in oxygen, but it'll still grow. That's facultative anaerobe, and that's what E. coli is. E. coli is a facultative anaerobe, and it can just switch from metabolic process to metabolic process as the need suggests. It's a pretty good trick. So here's our conclusion on the next couple of slides. Clostridium sporogenes, this is our obligate anaerobe. No growth in room air. Oxygen is toxic to this kind of organism, but it grows really well in the brewer's jar. It grows where there is no oxygen. In the thioglycolate tube, it's going to grow in the deepest part of the tube, deep, deep down in the anoxic region, no oxygen. Alkaligenes grew for us in room air and did not grow in the brewer's jar. We saw growth in the thioglycolate tube only in that oxic zone. And that tells us that alkaligenes is an obligate aerobe. It requires oxygen for survival. If you put it into a region with no oxygen, it cannot grow. Those are the two that are I think, the easiest ones to see in the results. The other two, not quite as easy. Enterococcus faecalis, this one grew in room air, and it also grew in the brewer's jar, but not as well. It grew into little colonies. It didn't grow very well. It grew in the lower oxic and anoxic regions of the thioglycolate tube, but the best growth was in the lower half of the oxic zone. And remember, we said that makes Enterococcus faecalis aerotolerant. And finally, we have our old friend E. coli, who grew in the room air, also grew in the brewer's jar, but not as well, and grew throughout the thioglycolate tube with the best growth all through the oxic zone. And this is classic facultative anaerobe. Now, there is one other category, right? There is the microaerophile, and we didn't get to see one of those. But if we had a microaerophile organism, what we would see is it would, and I've got this down at the bottom here for you. We would see growth in air, but it would be poor growth. Because remember, the microaerophile, it requires oxygen. It has to have oxygen, but it wants it in low concentrations. So it'll grow in air, but it won't grow well. It will not grow in the brewer's jar because it must have oxygen. It will grow only in the oxic zone and only in the bottom half of the oxic zone. So it has a very limited area for growth. A microaerophile has a very limited area where it can grow inside a thioglycolate tube. All right.
Can I ask a question regarding that?
Yeah.
So with the microaerobe, because of its limited area for growth, does that mean that there are probably less pathogens that come from that category?
Oh,
Or
that's
is
a
that
great question. That's a
Thank
great
you.
question. So we really can't relate oxygen category to the potential pathogenicity of an organism. You might think that, well, we know humans use oxygen, and we know we always have oxygen in our blood. And, if you think about the places where bacteria are, you would think that you could relate what kind of organisms make the best pathogens in us, but you really can't. And that's because you have to remember that we can get pathogens in all different locations in our body. For example, I'll give you a great example. Clostridium, just like Clostridium sporogenes, there are other Clostridium organisms that are obligate anaerobes. So you would think, oh, well, probably not going to be a human pathogen, right? There's a Clostridium organism called Clostridium tetani, which causes the horrible disease tetanus in humans. And the way you get tetanus is by being punctured by something, right? If you know anything about tetanus, people who step on, say, a rusty nail, or who somehow get a puncture wound that happens to have some of this pathogen on that device that punctured you, the cells are being driven down into your tissue where there's very little oxygen. And so they're able to grow, and they're able to divide, and they're able to cause disease. Other cells that are obligate aerobes, or otherwise using oxygen, if they get into our lungs or they get into our blood, they grow great. So we really can't look at oxygen category and know whether or not something is a good human pathogen, because there are all kinds of little nooks and crannies in the human body where any one of those categories could survive. It's a great question, though. It turns out pathogenicity is a lot more about whether or not an organism can evade the immune system. What makes a pathogen a good pathogen is whether or not it can evade the immune system, it can hide, or it can somehow beat the systems that are in place to find it and destroy it. Good question. Other questions? All right. So just like last time, a lot of material today, I know, but very important diagnostic tests for us when we're trying to figure out what we have in terms of microbial life and especially bacterial life. Very important diagnostic information to know what oxygen category something falls in. Like I said, most bacteria are facultative anaerobes, but when you find certain organisms that aren't, when you find the occasional microaerophile or you find an obligate anaerobe, it helps you narrow down what the possible identity of an unknown organism is. So it's quite helpful. Anybody have any other questions or comments or concerns? All right, very good. I'm going to let you go. Keep up the good work. And if anybody has any questions or they need me for anything, just give me a shout over Canvas. Okay?
Thank you.
All right. We'll see you next time.
Thank you.
Okay. Have a good night.